Hello and welcome back. And the next presentation is Abliva. And with us we have here today Ellen Donnelly, the CEO. And just as a brief introduction, Abliva is a Swedish biotech developing novel treatments for mitochondrial diseases. And with this, I'll give you the word now, Ellen.
Thank you. Hi, so I'm Ellen Donnelly. As I mentioned, I'm a CEO of Abliva. I've been with the company for 2 years. My background, I have a PhD in pharmacology from Yale. Then I went into management consulting, and I moved to Sweden in 2016, where I was the CEO of a sickle cell company. So excited to be here at Abliva. I've been at Abliva for 2.5 years now. So we're going to talk about a topic many of you probably don't know much about. It's mitochondrial disease. And so I'll talk about it a lot throughout the presentation, and feel free to ask questions at the end. We have our disclaimer. So we are focused at Abliva on building a company focused in mitochondrial medicine. And that may sound like a lofty goal for a small biotech company, but we have a lot of things that are in play that will help us do this. The first thing is that we have colleagues that have been working in mitochondrial medicine for over 2 decades. That is fairly unique in this area. So the mitochondria have kind of been, I think it's that untapped area of biology that many of us haven't really thought a lot about, which is pretty interesting considering they're in every cell in your body except for the red blood cells. We talk about neurons, we talk about cancer cells, we often don't talk about mitochondria. So today we're going to spend a lot of time talking about the mitochondria. We also have a research and development group. Again, rather unique. We have a lab at Lund University, which you can see here, focused on the science, and then we have a development group at Medicon Village in Lund, focused on the development of the assets. Also unique is that we have a portfolio of programs. We have one in late-stage development, so that is one in the study that will get it to the market if it's successful, and then we have another program that's ready for its first clinical study in healthy volunteers. We're publicly traded on NASDAQ Sweden in the small cap. So what are mitochondria? As I mentioned, they're in every cell in your body except the red blood cells. They're the powerhouse of your cell. They are responsible for generating the energy that your cell needs to do their job. That is very important. If you'll remember from your biology classes, they are generating ATP. And that energy is going on to do the cellular processes that each of these cells need to do. Now, in the organs in your body that need the most energy, they have the most mitochondria. So you can imagine if your mitochondria aren't working, those are the organs that are impacted most quickly. Muscles, your brain, your kidneys, they all have a lot of mitochondria, and so you usually see the issues will be in those organs first. The interesting thing about the mitochondria is they have different DNA than the rest of your body. Their DNA comes from your mom. We call it mitochondrial DNA, or sometimes you'll see mtDNA written. That's different than your nuclear DNA. And the reason that's interesting is it comes from your mom. So your mom may have the disease if you have it, but also the proofreading isn't the same as in the nuclear DNA. So there are often mistakes made. Depending on how severe those mistakes are, you may see an issue, you may not have an issue. So patients that suffer from mitochondrial DNA mutations or mutations in their nuclear DNA that affect their mitochondria, these are called primary mitochondrial disease. And they're rare diseases affecting about 1 in 5,000 individuals. They're devastating, debilitating disorders. They could lead to a shortened lifespan, or they could just really affect the activities of daily living. There are no approved therapies for systemic disease. These patients are diagnosed through a genetic test. They go to their doctor, they get tested, they find out they have a primary mitochondrial disease. There's nothing they can do. They're usually introduced to a patient advocacy group or a group of patients support group, and they're supported through the duration of their disease. And so if you go on our website, you can see a couple of patients. We have videos for both Rebecca and Roger, so you can see what the impact of this disease on their life. And then we have an interview also with Daniela on the website. But one of the examples we give, we have a disease or an asset called NV354. It's the second asset in the portfolio. And it's for a childhood disease called Leigh syndrome. And this one, I'll give you an example of a patient. So I'm a new mom, my son's 12 months old, right? Starting to develop normally like any child does. Suddenly gets a cold. And he doesn't seem to recover from that cold. The child just keeps getting worse. And mom's like, there's something wrong here. This wasn't a normal cold. I feel like my child's actually getting worse. Might go to see the pediatrician, and the pediatrician says, this is a bit odd. Come back if he doesn't do any better. Doesn't do any better, you come back, you finally get a genetic test and find out the child has Leigh syndrome. This is a genetic disorder, it's a primary mitochondrial disorder. That child is gonna go downhill very, very quickly. Average lifespan, maybe 5 years, 10 years. They'll start to lose all of their motor milestones. start to be fed by a feeding tube, and maybe in bed, and unable to get out of it until they pass away. Can you imagine being that parent? I mean, they thought they had a healthy child. The child got sick, and suddenly you find out this is a death sentence for your child. Those are the kind of disorders we're working on. This is very serious medicine that we're doing. KL1333 is the lead asset. So this is targeting adults that have a primary mitochondrial disorder, and they also, in that disease, they suffer from fatigue and myopathy. Myopathy is muscle weakness, and what we see with these patients is they have a hard time initiating movements, a hard time getting up and standing up and sitting down, and then the fatigue is overwhelming fatigue. We say that we'd like to call it mito fatigue because it isn't the fatigue you and I feel. It's really the substantial fatigue that, I have to plan my shower because a hot shower actually takes so much energy. I can't do anything after I take that shower. So the lead asset, again, adults with fatigue and myopathy. That's in a pivotal study. We'll talk about that right now in a few slides. The second asset, NV354, this is the one for the kids. This is different. It gets into the brain, whereas the first asset doesn't. And this one's ready to go into phase 1 studies, and early programs exist as well. So why haven't you heard about these before? I think that this is really we've had a confluence of activity within this space that have really made this the time to go after these diseases. The first thing that really has happened is we have an increased scientific understanding of the space. Mitochondria are complex organelles. This is a heteroplasmic disease, so some are affected. You might have in a muscle cell some mitochondria that are affected, some aren't. And then that muscle cell may be affected, but maybe kidney cell isn't. So it's been a little bit of a complicated path forward to figure out how to actually target this disorder. A nice thing about this area is it's a rare disease, so we do have the regulatory tools that are available to other rare diseases. I'm sure you're all aware of orphan drug designation. This allows you to have market exclusivity for 7 or 10 years in the US and EU. An important tool because it allows us to put the money into the development of the drug knowing that we will have market exclusivity when it gets out. We have that designation for both of our assets, KL1333 and NV354. Another opportunity is obviously pediatric rare disease voucher. You can get this and that can be sold and give you some opportunities there. A fast track designation is the FTD, and then breakthrough therapies. These are all available to companies working in this space and tools that we hope to take advantage of. Something that's happened recently as well is now that we are starting to diagnose these patients more frequently because they get identified, they get sent for genetic testing, and genetic testing is available in most countries throughout the world. So they're getting diagnosed. There's centers of excellence that are coming up. And these centers of excellence have physicians that are interested in both the science and the development of drugs for this area. And so you're seeing this confluence of people focused in this disease. And so in some states or countries, we actually have one center. And all of the patients that are diagnosed with this disorder will be sent through that center, tracked by that center. This helps us a lot when we're trying to recruit studies because we know how many patients are seen by that study, this study site, and then we hope to recruit patients from that group of individuals. And in some cases, if a whole country has one center, you can imagine it's a lot of patients that we're looking at recruiting. It also helps us with the sales once the drug is ready to go to market. So we have centers of excellence where we have this convergence of all these patients, and then we also have these patient advocacy groups. And this is a bit different, I think, in this area because they seem to have consolidated. An example, I think a counterexample of this would be DMD in the US, where you have a lot of patient family groups that put $1 million behind a therapeutic. Here we've really had consolidation of these patient advocacy groups because they understand that, if they can put more money in the pot, it's obviously going to be more helpful. And so that is very helpful to us. UMDF, a good example, is the leading patient advocacy group in the US. They helped us recruit our patient study. They have resources for the patients online. They're constantly in the discussion, helping us find patients, attract patients to the study, and really bring therapeutics to this important group. So that's all happened in the last few years. And then I think as a company, we have a very strong internal position. We do have 2 first-in-class assets. That makes it a little more difficult, but it also means that we can be to the market and take a strong position. We're in line with the competition. We're all in the same pivotal space, and there aren't many players, actually. KL1333 has about 3 competitors, and we're all complementary. So actually the word competitor, I guess, doesn't really factor in here because we could all be approved and be used in together. And then MV354 has a similar number. So not a big competitive landscape here. And we also have a strong patent estate. That's been one focus of the company, is really ensuring that our assets are protected. So all of this has kind of, I think, come together to really make this a strong opportunity for Abliva and for the industry in this area. I think what it says is it's decreased risk. We have decreased time to market because of these incentives, and we have decreased cost to market. So that all makes for a nice package and an increased commercial opportunity. So here you can see the peak sales projections that we have this is for the lead asset for KL1333. This is a pretty, I call it a back of the envelope exercise. So what we did here is we said, what do you have to believe to hit $1 billion in peak sales? And it's a pretty easy estimate, right? You have the prevalence of the disease, which is about 60,000 in the US and Europe. And then you say, okay, I'm gonna assume 50% of those are diagnosed, because we still have a problem getting patients diagnosed. They might not get sent for that genetic test until a year or two afterwards. So we'll assume 50% are diagnosed, 50% are treated. And that's in a great scenario where you actually have a therapeutic that's available to treat them. And then we said we were second to market. Price it at about this, $160,000 per year. Again, a pretty conservative estimate. Right now, a similar therapy in the US is about $300,000. And we've had that confirmed with payers that would probably be the price that they would put this drug at. But we don't know what's going to happen with pricing, especially in the US. There are lots of discussions ongoing about pricing of rare therapeutics in the US. So to be conservative, we took $160,000 per year, split that in half for Europe. You can see it still hits $1 billion opportunity. So really big commercial opportunity for the lead asset here. So what is this? What is this lead asset? So it's a modulator of two coenzymes in the mitochondria. These are NAD+ and NADH. Fairly certain none of you remember about these coenzymes. The reason they're important is because they need to be at the right ratio in order to generate ATP. If they're not, in patients with mitochondrial disease, they're just producing a ton of NADH. And their electron transport chain doesn't work, so they can't convert it to NAD+. So the system has a whole bunch of this enzyme that they can't do anything with. So the enzyme doesn't help us, we don't get energy. Simple story, right? So we're modulating the amount of these two enzymes so that they're in the right ratio. Interestingly, we've dosed over 110 healthy volunteers and patients with the drug and have seen no safety issues. So no SAEs out of a number of studies. So we have a really strong safety profile, and we have signals of efficacy. We're gonna show you that data in a minute. But that's important. So we've dosed patients with our drug and seen good effects. I'll show you those. We have this potentially registrational study ongoing. People often ask why I keep saying potentially. Like, why are you modulating? Because we don't know the data yet. So we have discussed with the regulators the fact that this study, if positive, could be registrational. But until we have the data, we don't know. So we like to put potentially before it. As I mentioned, limited competitive landscape here. All of us are at the same stage. And we have this strong patent estate with the ODD designation. So I mentioned data. Everyone likes to see the data. And unfortunately, this is the only, I think I have another data slide, but we have one slide of data. So what we did here is we put this in patients. So this was a 10-day study, and the patients were dosed with only 50 milligrams of drug. So we wanted to go in, it's our first experience in patients, so we want to make sure that they don't have any issues. So we went in with a low dose, 50 milligrams. In this study, we had 3 clinical endpoints. 2 of them were fatigue, and 1 of them was myopathy, because we knew that those were the symptoms we wanted to look at in this pivotal study. We also knew that these are the symptoms that the mechanism is likely to address. So we wanted to look at 3 endpoints. So we had 2 fatigue scales and a myopathy scale. And then we looked at them. We had 2 patients on placebo. This looks like the drug. Everybody was blinded, double blind, closed, not an open label study, which you often see in this area, but this was a legitimate placebo control. 6 patients got the drug. And you can see here, the gray is the placebo patients. KL1333 is in black. There's a big difference between those 2 groups. 2 of them are going up, 6 of them are going down. That difference, although not statistically significant because this is too small a cohort, that difference we believe is clinically meaningful. So what that means is a patient should be able to feel that difference. The only way we know that is because you look at different indications or different TAs where this has been used, and it does look like about a 3 to 5 point difference is clinically meaningful in different patient populations. So that was nice data. The other fatigue scale looks very similar to that. That was This was a 7-day recall. That was a 24-hour recall. This test is a different kind of test. This is muscle function. So this is called the 30-second sit to stand. So basically, the patient has to sit down and stand up as many times as they can in 30 seconds. We count it. It's a number. And you can see here that there's about a 2-point difference in that. So they were able to do this 2 more times in 30 seconds. Try it. It doesn't sound like a big number. It's actually a pretty significant difference when you're doing that test. And here you can see, again, a good difference between the 2 groups. Again, in other disease areas, this was clinically meaningful. That's important. Patients can feel the difference. Patients could feel the difference after 10 days at a low dose of this drug. We have other data that shows that we had an exposure-effect relationship. The reason this is important and what it means is that if a patient got a lot of drug, had a lot of drug in their body when we did the assessment, they did better. So this suggests that it's actually the drug that's driving the effect. As you know, sometimes you get off-target effects. You get effects that just happen because it's time of day. These are actually, we believe, driven by the drug, and we have evidence to show that. The other way you show that is through target engagement. So in this study, we have lactate and pyruvate. We use those as our biomarkers, and we saw a good dose-dependent effect on lactate pyruvate when we dosed our drug. So we call that target engagement, and that was an important thing as well. So an interesting experience in patients, super happy with the data. We hit on all 3 clinical endpoints. We showed the exposure-effect relationship, and we also showed target engagement. So that data gave us the confidence to move into the study that we're currently in. This is the study. It's called the Falcon study. We feel like we're giving the patients wings to fly, and hopefully they'll get over their fatigue, they'll get more functioning in their muscles and be able to do more in their daily life. It's a double-blind, parallel group, randomized study. So we've got the normal design here. It's in adults with genetically confirmed disease that have fatigue and myopathy. And right now, they're in a screening period. So what happens, the first thing that happens is a patient comes in, we think that they might be relevant and eligible for the study, and they're tested, they're genetically confirmed, and then they're tested for fatigue and myopathy, because you want to make sure those levels are high enough so the drug can actually do something. So that's our 8 to 12-week screening period. Once they get through that and they show that they maintain this fatigue and this myopathy for that period, they're treated with the drug, and the treatment goes for 48 weeks. One thing that's really important in this disease is these patients are not in good shape. You can imagine that they don't want to come into the clinic every month and be tested. This is a long study. We're trying to register a drug on this, so it needs to be long. We need to get the safety profile, but we can't bring them into the clinic. And so this is basically, it's done at home. So the patients will take the drug in the morning, they take the drug at night, and many of the assessments are done at home. We have home healthcare nurses that come when necessary. And we have a travel program, so when they do have to go into the clinic, they make one phone call and we get them there. Right now, we are running the first 40 patients to an interim analysis. So what does that mean? Right now, we're recruiting patients into the study. They're going to run the full year, but at 6 months, once we have 40 patients that have hit about 6 months of treatment, we'll do an interim analysis. In that, it's blind, but we look at the power of the endpoints and we look at the futility of the study and see if it needs to go forward and how big the sample size will be going forward. And so that's an important time to get an understanding for the power of those endpoints. And you can see the study will end up being 120 to 180 patients in total. One of the other things that we've done to de-risk the study is we have an alternate primary approach. And what does that mean? So we have 2 primary endpoints, and they're actually analyzed independently. So either one hits, we have is statistically significant, we have a positive study. It's a unique design. Usually when you have 2 endpoints, they're primary, they're co-primaries. They need to move together. It's completely different, alternate endpoint approach. They move independently, and you assess them independently. So basically what we say is we have 2 shots on goal. Either one is statistically significant. We have a reason to go to the regulators and ask for approval of the drug. We also have a number of secondary endpoints and exploratory endpoints that will support that. We're running the study in the US and EU, and we have lots of updates available online. You can either go to our website, and you can see all about the clinical study there. You can go to clinicaltrials.gov. But again, ongoing study. Interim analysis is expected in the first half of next year. Second drug is earlier, so I'm not going to spend so much time on this, but this is interesting because it came out of our labs. It's a prodrug of a natural compound called succinate. Succinate is used to create energy within your mitochondria, and we're just giving the cells more of that. It's hard to get into the cell, so we had to make a prodrug technology. So the prodrug gets it into the cell, and then we get the energy from the succinate. This targets diseases with complex 1 disorders, so different patient population here. We've got good drug properties, again, nice patent portfolio and ODD designation in the US, and it's clinic ready. So here I just show some data. We showed improved function in Lee patient cells as the first graph. The second graph shows the nice brain distribution. This is really important because we're targeting the CNS in this drug. And then you can see the mouse model in this is quite nice because they actually just affect complex 1. So this is kind of a translational model because it's impacted the exact problem we have with the complex 1 deficiency. So in these NDUFS4 mouse model, you can see the motor activity decline, and it's fixed, and then the weight improves in these animals. So this is the team. As I mentioned, we're based in Lund. I'm based in the US subsidiary out of Boston. So you can see me. Katarina Johansson is our CFO, Magnus CMO. Eskil is the founder of the company. He's the CSO. He's the one that's running the lab at Lund University. And then Dag just joined us. He is our head of clinical operations and responsible for the study. But the really important people here are obviously the scientific advisory board. And I know you probably hear this from every rare disease company, but we cannot do it without our advisors. These guys are the experts in the area, and so you can see them here. We're excited because we're going to see most of them. The big mitochondrial meeting is every 2 years, and it's happening next month in Bologna. So we're going to have a chance to meet with our SAB at that time, other patient organizations and physicians involved in this area. So that'll be a fun meeting. So that's it. That's our story. We're really interested in becoming a global leader in this area. I think we have all the components to do that. And we're just really right now focused on operational excellence in this study to make sure that we can get to that interim analysis and get some data. So that's Abliva. Thank you.
Thank you very much, Helen, for the presentation. Very nice. A few minutes for questions now. And I will start asking you about your Falcon study, your phase 2. If I understand correctly, you're only targeting adults for that study. How come that you did not also include like children, for example? I mean, what is the average age of diagnosis of these patients? Is it that it only happens in the adult life, or?
Right. So primary mitochondrial disease, there are a number of different diseases within that category. And so what we're trying to do is find a homogeneous population, but yet a big enough population to make a real difference in the group. And so what we've done with this population is we've focused on a number of genetic mutations. So we're looking for specifically M3243 alanine to guanine, and that tends to be a rather large population. So we're looking for the genetic background, and then we're looking for the phenotypic background. It's usually normal that you would start in an adult study before and that's usually step 1 is the adults, and step 2 is the pediatrics, and we could do that next. But we felt that this was the biggest label and the most broad label to start with the adults first.
Okay, no, thank you. And a second question would be on diagnosis. I mean, you're saying that, of course, it's underdiagnosed where we are now, and this should increase in the future. Where are we there in terms of diagnosis? I mean, and how hard would it be to make it into guidelines? Because that's ideally, in an ideal world, everybody would be at some point with specific symptoms.
Absolutely. I mean, the nice thing about this area is it's a genetic test. And it's readily available in many countries. Interestingly, in Sweden, one of the confounding factors in this disease is they often have diabetes, and we call it diabetes 1.5 because of the pancreas involvement of the mitochondria in the pancreas. And so in Sweden, they'll actually test a patient. If they're testing them for diabetes, they also test them for primary mitochondrial disease. So it's one of the tests that's done when they're going in for diabetes. So it is becoming much more common that they'll be diagnosed. And as the literature starts to come out about this disease, and there's been a huge increase in the amount of literature too, we've seen increased diagnosis. I think there's also an ability for the physicians have started talking about it more clearly, because the concern originally was that you might have one issue that takes you to a hearing specialist. And it's not until you have a second issue that takes you to a neurologist that they say, oh, there's something going on here. It might be mito disease. Let's test them. Now it's being picked up much more quickly. It's not taking that 2 or 3 years to actually get it diagnosed. But I think as therapies start to be identified too, as we get a drug approved in this area, that's going to increase the diagnosis a lot. Because you can imagine being a physician, you're like, well, I can diagnose this patient with M3243, A to G mutation called MELAS mid-spectrum, but what are they going to do with that information? So that's difficult as well. So I think with the increased approval of drugs in this area, will increase the diagnosis rate as well. It's getting much better. I think in the it's logarithmic growth at this point.
That's great to hear. And another question, going back to your Falcon study, you mentioned that you have this alternate primary endpoint. You can hit both or one in order to be approved. One of them, if I recall correctly from your slide, it's validated. The other one is not validated, or what's the situation?
Well, they're both validated. So the one is 30 seconds sit to stand. This is the one people have probably heard of, where you're standing up and sitting down as many times as you can within 30 seconds. It's also called the sit to stand test, or the there are lots of different versions of this test. So that's been used across therapeutic areas. The fatigue scale is a little bit different. So what we did with that is that there's a fatigue scale. There are many fatigue scales that are used across therapeutic areas, but they tend to be pretty general. It's like putting a Band-Aid on a cut. Hopefully it'll work. But they're not specific to that disease. So you're just hoping these questions that you're asking, these 8 questions, may be relevant to the disease. And so when we went to the FDA, we said, we want to test fatigue, but we're not happy with this generic approach. What can we do? And so we spent the last 2 years validating our fatigue endpoint. What does that mean? So what you do is there are a bunch of questions that exist in a fatigue database that have all been validated. They're all official questions. And you test those with the patients, and you say, what does this question mean to you? Is this question relevant to your disease? Do you understand this question? Because that's a lot of the problem, is that people either don't interpret the question the same, or they don't understand it. In this case, one of the questions was, in the past 7 days, how often have you had an issue doing strenuous exercise? Now, our patients don't exercise. They can't, in many cases, can't get off the couch. So to ask them about strenuous exercise is a wasted question, and frankly, the reason a lot of these fatigue scales don't work. And so we tested this. We did concept elucidation and validation with patients over 2 years, and came up with a set of 8 questions that are relevant, I think it's 9, relevant to the patients and relevant to their disease. And so we'll be publishing that this next month at the big mitochondrial disease medicine. So both endpoints are validated. It's just we have a little bit more work to do with the fatigue scale as we do some in-study parameters to make sure that it's robust.
Great. And one last question. Your Falcon study it's a quite long study. It's a 12-month treatment. How well are you now in terms of cash position? And how does it look, your cash burn in the next quarters?
Right. Yeah, good question. I didn't bring that up here. So we raised 200 million SEK last June to get us to the interim analysis. And so that's still the plan. So good cash position. Happy to have that. And we brought in, the nice thing about that raise was we did it at an extremely difficult time in the market. The Ukraine war had broke out. The market we thought was at a low. I guess maybe it wasn't. But it was still not a great time for the market. And we brought in new investors, and we did it at a very low discount. So really happy to have fresh blood in the company and have their backing as we move to this next stage. So that was a nice round.
Great. Thank you very much, Helen, for being here today with us and for the presentation.
Thank you.